The cells which are closely associated with sieve tube elements are nothing but companion cells. Companion cells are special parenchyma cells. So, companion cells having protoplasm till its death. So, the companion cells are associated with sieve tube element. Both are interconnected with one another through pit fields. Where the pit fields are located? Present on their longitudinal walls. What is the function of companion cells? The nucleus of the companion cell maintain the pressure gradient of the sieve tube element. The functioning of the sieve tube element is also controlled by companion cells. So, companion cells having two functions. The functioning of the sieve tube element is controlled by companion cell, and the pressure gradient of the sieve tube element is maintained by companion cells. Next, phloem fibers. Phloem fibers. What is phloem fiber? A sclerenchyma cell present in phloem. Sclerenchyma cell present in phloem, that is called as phloem fiber. Phloem fibers are long elongated cells. With tapering ends. They are elongated cells with tapering ends. Generally, phloem fibers are unbranched cells. That is also an important point. They are unbranched cells. They are elongated cells with tapering ends. In phloem fiber, the cell wall is quite thick. Made up of lignin. In mature phloem fibers, the protoplasm is degenerated. The protoplasm is degenerated in mature phloem fibers. So then they become dead cells. The mature phloem fibers are dead due to degeneration of the protoplasm. Young cells having protoplasm, the mature cells are without protoplasm, so may be treated as dead cells. Next, another very important point, phloem fibers are absent in primary phloem. They are absent in primary phloem, but present in secondary phloem. Present in secondary phloem. That is important point. Phloem fibers are absent in primary phloem, but present in secondary phloem. Secondary phloem fibers may also be called as bast fibers. Secondary phloem fibers bast fibers. The phloem may also be called as bast. The another name for phloem that is called as bast. The phloem fibers may also be called as bast fibers. Example for bast fibers, jute fibers, flax fibers, and also hemp fibers. Jute fibers, flax fibers, and hemp fibers are the example for secondary phloem fibers. So jute fibers are obtained from secondary phloem of the plant, Carcorus. Secondary phloem of the plant, Carcorus. Flax fibers are obtained from the secondary phloem of the plant, Linum. Linum is the name of the plant. Hemp fibers are obtained from the secondary phloem of the plant, Cannabis. The secondary phloem fibers of cannabis, that is called as hemp fibers. The secondary phloem fibers of the plant Linum, flax fibers. The secondary phloem fibers of Carcorus, called as jute fibers. That is about fibers. Phloem fibers are long elongated cells with tapering ends. The cell walls are quite thick. The mature phloem fibers are without protoplasm. Then they become dead cells. Phloem fibers are absent in primary phloem, present in the secondary phloem fibers may also be called as bast fibers. Example for bast fibers, jute fibers, flax fibers, and hemp fibers. The longest fiber is rami fibers. Rami fibers. So that is about fibers. Next, phloem parenchyma. Phloem parenchyma. They are elongated cylindrical shaped cells with tapering ends. They are elongated cylindrical shaped cells with tapering ends. Whose cells are cylindrical shaped in complex tissue? What are cylindrical shaped cells? That is phloem fibers and also in xylem vessels. Vessel member is cylindrical shape. These two are cylindrical shaped cells. Vessel member in xylem vessels and phloem parenchyma cells. They are elongated cylindrical shaped cells with tapering ends. Cell wall is thin or thick? Thin, made up of cellulose. The cell walls are thin and are made up of cellulose. The cells are living or dead? Cells are living. Having vacuolated protoplasm. The cells are living having vacuolated protoplasm. The phloem parenchyma cells are connected with pits. through pits present on their cell walls. So the cell wall having pits. The cell wall having pits. These are the cells. These are the pits. The cell wall of the phloem parenchyma cells having pits. Through these pits, plasmodesmata connections are present. This is plasmodesmata. The cell wall having pits through which plasmodesmata exist between the cells. The plasmodesmata exist between the cells. What is the function of plasmodesmata? Helpful for translocation of materials from one cell to another cell. Translocation of materials from one cell to another cell. If materials are translocated from one cell to another cell, it is called as plasmodesmata. What are plasmodesmata? Cytoplasmic strands. Plasmodesmata is nothing but cytoplasmic strands. It connects the two adjacent cells and helpful for translocation of materials from one cell to another cell. That phenomenon is called as translocation of material from one cell to another cell. Intercellular transportation. Intercellular. If translocation takes place within the cell, intracellular. Translocation takes place within the cell, intra. From one cell to another cell, intercellular. So plasmodesmata plays an important role in intercellular transportation of materials from one cell to another cell. Intercellular transportation. The phloem parenchyma cell also store food materials. It also stores food materials. And ergostic substances like resins, latex, and mucilage. The phloem parenchyma cells also store secondary metabolites like resins, latex, and mucilage. But xylem parenchyma cells store tannins. Xylem parenchyma store tannins, but phloem parenchyma cells store resins, latex, and mucilage-like compounds. They are not the food material. They are the waste material, that is ergostic compounds or secondary metabolites. So that is about phloem parenchyma. The phloem is composed, in angiosperm, the phloem is composed of how many kinds of cells? 4 kinds of cells, that is sieve tube elements, companion cell, phloem fibers, and phloem parenchyma. What is the difference between sieve tube element and sieve tube? Sieve tube and sieve tube element. Sieve tube element is a single cell. Sieve tube is nothing but a tube formed by the union of sieve tube elements. Sieve tube is nothing but a tube made up of many sieve tube elements. A single cell of sieve tube is called as sieve tube element. Next, the end walls of the sieve tube elements are perforated or non-perforated. End walls of the sieve tube elements are perforated in a sieve-like manner called as sieve plates. They are perforated in a sieve-like manner called a sieve plate. The pore present in between the sieve plate, sieve pore. The translocation of food materials takes place from one sieve tube element to through another sieve tube element through sieve pores of the sieve plates. Translocation of food takes place from one sieve tube element to another sieve tube element through sieve pores of the sieve plate. When the injury occurs in sieve tube elements, what is secreted? P protein was secreted that act as a plug and completely block the sieve pores of the sieve plate. In older sieve tube elements, what is secreted? Callose. Callose is a carbohydrate, not protein. That callose act as a plug, completely block the sieve pores of the sieve plate and obstruct the conduction of food materials. When the sieve tubes are older, what is secreted? Carbohydrate was secreted. That is nothing but callose. When the sieve tube shows injury, P protein was secreted. That also block the sieve pores of the sieve plate. Next, companion cells and sieve tube elements both are linked with one another by pit fields. Where they are located? Their longitudinal walls. The mature sieve tube elements are with nucleus or without nucleus? Without nucleus, having only peripheral cytoplasm with a large vacuole and nucleus is absent. Though the nucleus is absent, mature sieve tube elements may also be treated as dead or living cells. Living cells. Phloem fibers are absent in primary phloem, present in phloem fibers are branched or unbranched fibers? Unbranched fibers. Phloem fibers may also be called as bast fibers. Example for bast fibers: jute fibers, hemp fibers, and flax fibers. Flax fibers. Next, phloem parenchyma. The phloem parenchyma cells are cylindrical shaped elongated cells with a tapering end. The cell walls having pits. Through these pits, plasmodesmata exist between the cells that helpful for translocation of materials from one parenchyma cell to another parenchyma cell. What is stored in parenchyma cells? Food materials and also ergostic compounds like resins, latex, and mucilage-like compound. Next, another important point in phloem parenchyma. Phloem parenchyma is absent in the phloem of monocots. In monocots, in phloem parenchyma is absent. Very, very important point. In phloem of monocots, phloem parenchyma is absent. So that is about complex tissue. The phloem is of two types. That is primary phloem and secondary phloem. So primary phloem is formed from anybody? Procambium. Very good. Primary phloem and primary xylem both are formed from procambium. But secondary phloem is formed from? Yes, vascular cambium. Not fascicular vascular cambium. Fascicular vascular cambium is a primary meristem. That is intrafascicular cambium, may also be called as fascicular vascular. This is only vascular cambium. It is formed by the union of intrafascicular cambium and interfascicular cambium. Next, a primary phloem having two kinds of cells. That is protophlloem and a metaphlloem. Primary phloem having two kinds of cells, protophlloem and metaphlloem. Protoploem having narrow sieve tubes. Narrow sieve tubes, not sieve tube elements. Protoploem having narrow sieve tubes, but metaploem having bigger sieve tubes. Protoploem of primary phloem having narrow sieve tubes, metaploem of primary phloem having bigger sieve tubes. So that is about complex tissue. Next are tissue systems. Tissue systems. Based on structure and location, tissues are divided into three types. Tissue systems are divided into three types based on structure and location. Based on structure and location, tissue systems are divided into three types. One is epidermal tissue system. Second one, ground tissue system. And third one, vascular tissue system. Like that of three kinds of systems: epidermal tissue system, ground tissue system, and vascular tissue system. Epidermal tissue system constitute epidermis, cuticle, stomata, and epidermal hairs. So epidermis, cuticle, stomata, and epidermal hairs are included under epidermal tissue system. Epidermal tissue system constitute four kinds of structures, that is nothing but epidermis, cuticle, stomata, and epidermal hairs. First, epidermis. Epidermis is how many layered in thickness in most of the plants? It is single layer. It is mostly single layer. But in some plants it is multi-layer. In some plants the epidermis is multi-layer. Multi-layered epidermis is called as multiple epidermis. Multi-layered epidermis may also be called as multiple epidermis. Multiple epidermis is present in epidermis of velamen root. The epidermis of the velamen root is having multiple epidermis. And epidermis of nerium leaf. The epidermis of the nerium leaf is also multiple, having multi-layered epidermis. Epidermal cells are elongated cells. They are elongated cells and are compactly arranged, closely arranged. Compactly arranged cells and found in the form of continuous layer. The cells are elongated, the cells are compactly arranged and are found in the form of a continuous layer. Like this. These are the epidermal cells. They are elongated cells and are closely arranged in the form of a continuous layer. They are found in the form of a continuous layer. The cell walls are thin, made up of cellulose. The epidermis is made up of parenchyma. The epidermis is made up of parenchyma. What is the character of parenchyma cells? The cells are living having vacuolated protoplasm. The cells are living with vacuolated protoplasm. The cells are living with vacuolated protoplasm. So this is vacuole. And this is cytoplasm along with the nucleus. This is nucleus. So that is about epidermis. Generally, epidermis is single layer, but some are having multi-layered epidermis, which may also be called as multiple epidermis. Multiple epidermis can be observed in the epidermis of the velamen root and epidermis of the nerium leaf. But in most of the plants, the epidermis is single layer. The epidermal cells are elongated and are compactly arranged and are found in the form of a continuous layer. They are parenchymatous having vacuolated protoplasm, having protoplasm along with a centrally located large vacuole. The cells are living or dead? Living because of having protoplasm. So that is about epidermis. Next, cuticle. What is cuticle? A thick waxy layer present above the epidermis. A thick waxy layer present above the epidermis, that is called as cuticle. A thick waxy layer present above the epidermis is called as cuticle. Cuticle is thin or absent on the epidermis of hydrophytes. Cuticle is very thin or absent on the epidermis of the hydrophytes. Cuticle is moderate in mesophyll cells. In mesophyll cells, the epidermis having moderate cuticle. In xerophytes, the epidermis having thick cuticle. In xerophyte, the epidermis is very thick. That helpful to prevent the loss of water by transpiration. But in all roots, cuticle is absent. There is no cuticle on the epidermis of the root. But in hydrophytes also, cuticle may be absent or present. If it is present, it is found in the form of thin film-like. That is very thin layer. But mesophyll cells having moderate epidermis cuticle, but xerophytes having thick cuticle, but in roots cuticle is generally absent. What is the function of cuticle? Prevent the loss of water by transpiration. Cuticle prevent the loss of water by transpiration. This is cuticle. This is epidermis. It is helpful to prevent the loss of water by transpiration. Next, the third one, stomata. Third one, stomata. Where stomata are located? Stomata are commonly present in the epidermis of the leaves and also the epidermis of young stems, not older stems. The epidermis of the leaf and epidermis of the young stems having stomata. The epidermis of the leaf and epidermis of the young stems having stomata. What is the function of stomata? Regulate the rate of transpiration. And exchange of gases. Regulate the rate of transpiration and exchange of gases. That is the function of stomata. Stomata has two guard cells. Generally, stomata having how many number of guard cells? Only two. This is guard cell. In between the two guard cell having a pore. That is called a stomatal aperture. It may also be called a stoma. Stoma means stomatal pore. It is present between the two guard cells. Stomatal aperture or stoma. In dicot leaf, what is the shape of the guard cells? Bean shaped or kidney shaped. In dicot leaf, bean shaped or kidney shaped guard cells are present. So this is bean shaped guard cells. In a bean shaped guard cell, the outer wall is thin or thick? Thin. And the wall present towards pore side is thick. This is thick wall. And this is thin wall. The outer wall of the guard cell is thin, and the inner wall of the guard cell present towards the stoma side is thick. Next, what is the shape of the guard cells in monocots? God cells are dumbbell shaped. This is dumbbell shaped God cells. The God cells are dumbbell in shape in monocots. In dumbbell shaped God cell, the middle walls are thick. These are the middle walls which are thick. The thick middle walls, and the end walls are thin. The end walls are thin and middle walls are thick in dumbbell shaped God cells. Next, God cells are the only epidermal cells having chloroplast. God cells are the only epidermal cells having chloroplast. No other epidermal cell having chloroplast except a God cell. God cells are also the epidermal cells, but no other epidermal cell having chloroplast except God cells in higher plants. So what is the function of God cell? Due to having chloroplast, they regulate the process of opening and closing of stomata. God cells regulate the opening and closing of stomata. Due to having chloroplast, they regulate the opening and closing of stomata. When stomata opens, when the opening of the stomata takes place, when the God cell becomes the turgid, when the God cell becomes the turgid, stomata opens. Turgid means, what is turgid? Absorbs water and becomes swell. God cell absorbs water and becomes swell. That is called as a turgid cell. When God cells loses turgidity, when the cell loses turgidity means it becomes a flaccid. Closure of stomata takes place. When God cell becomes a flaccid, flaccid means loses water, closure of stomata takes place. When God cell becomes turgid, that is gain water, opening of stomata takes place. So opening and closing of stomata is controlled by the turgidity of the God cell. Opening and closing of stomata is mainly controlled by the turgidity of the God cell. When God cell becomes a turgid, stomata opens. When God cell loses turgidity, closure of stomata takes place. Like that, God cells are the only epidermal cells having chloroplast. But in hydrophytes, all epidermal cells having chloroplast. In hydrophytes and also in bryophytes. In hydrophytes and also in bryophytes, all the epidermal cells having chloroplast and perform photosynthesis. All the epidermal cells also having chloroplast in sciophytes. Sciophytes mean shady plants. The plants growing in shady localities. They are called as sciophytes. All sciophytes also having chloroplast in all epidermal cell. The sciophytes, the hydrophytes and bryophytes having chloroplast in all epidermal cell. But in higher plants, only the God cell having chloroplast. In higher plants, only God cell having chloroplast. No other epidermal cell having chloroplast except God cell. So that is regarding stomata. Next, another one, subsidiary cells. What are subsidiary cells? They are also special epidermal cells like that of guard cells. But what is the difference between guard cells and subsidiary cells? The subsidiary cells, though they are epidermal cells, they differ with the guard cells in their shape and size. They differ with the guard cells in their shape and size. Some epidermal cells in the vicinity of the guard cell are specialized in their shape and size. Such as specialized epidermal cells which differ with guard cells in their shape and size are called as subsidiary cells. What is the another name for subsidiary cell? Accessory cells. Some of the epidermal cells in the vicinity of the guard cell. In the vicinity of the guard cell means differ with the guard cells in their shape and size. Such kind of epidermal cells which differ with guard cells in their size and shape are called as subsidiary cells. The number of subsidiary cells are 2 too many. The number of guard cells are only 2. Number of subsidiary cells are 2 too many. Guard cells, subsidiary cells, and stomatal aperture. These three together called as stomatal apparatus. The two guard cells and the subsidiary cells and stomatal aperture together called as stomatal apparatus. So that is regarding stomata. Next, epidermal hairs. Yes, continue tomorrow. Epidermal hairs.